Hello, Bio 104 students. In this lecture, we're going to be covering evolutionary radiations, looking at rates of evolution, rapid radiations, and drivers of extinction. Now, already in this class, we have, in several instances, touched upon lineages that are exhibiting differential rates of evolution. Remember early on talking about Darwin's finches, we talked about how populations can rapidly change their characters in response to natural selection. We talked about speciation, patterns of sympatric speciation in freshwater sticklebacks in lakes in British Columbia, the rapid evolution of limnetic and benthic forms. When we started talking about animal evolution, we explored the Burgess Shale fauna, and we talked about the rapid radiation of the major lineages of animals and how that radiation was not only rapid, but was characterized by the evolution of appreciable disparity among these different lineages. And we've talked about mass extinctions that have happened in very short intervals of time, such as the end Cretaceous extinction at about 66 million years ago that led to the extinction of the dinosaurs. Now, the question is, other than we've talked about utilizing the fossil record to detect periods of mass extinction, but you may be asking, and in fact, several of you have asked in the course of our discussion groups, how do we really know about differential rates of evolution? And several you students have been asking, how do we estimate those time-calibrated phylogenies that we've shown a couple of times in the course? Well, the first thing to keep in mind is that when we're looking at phylogenies the way we looked at them up to date, phylogenies are only giving us a sense of relative timing. For example, the example here, all we can say is that A happened before B and C. However, we can't really say, given this phylogeny, the difference in timing of B and C. That is, C happened before B or B happened before C. Even though C in this clade is phylogenetically more nested than B is in this clade, the fact that they're in different clades and there's no sense of absolute timing in this phylogeny, we really can't say anything about the timing of the events depicted in the phylogeny with regard to nodes B and C. All we could say in this case that indeed A happened before B or C. Now, when we do have information about absolute timing, as is shown here in this time-calibrated phylogeny that we call a chronogram, meaning that it's relating to time. So here the branch lengths are drawn to reflect the estimated time, in this case of millions of years before the present. So now if we have this time-calibrated phylogeny, so remember without age information, all we could say is that A happened before B and C, but indeed in this case, we could see that the common ancestor depicted at node C is estimated to be older than the common ancestor depicted at node B. Now, how do we get these absolute dates? Well, the answer really comes down to two lines of evidence, or two approaches. One is the fossil record. Fossils, if they exist, we could place them into the phylogeny, and this provides a minimum age for clades. But then the question becomes, how much further back in time is the age of the clade relative to that minimal age implied by the fossil? Now, when we're estimating divergence times using molecular data, we're relying on what is called a molecular clock. And the molecular clock is the observation that here we're looking at, say, the percentage of amino acid changes in the hemoglobin gene versus the time that the two lineages separated, or the age of their common ancestor. And in this case, we see this correlation in that the older the evolutionary age between the lineages, the more amino acid changes there are at the hemoglobin. Now, this molecular clock insight was introduced in 1962 through a study of these of hemoglobin amino acid sequences. So, and this is a table from their paper, Zucker, Kendall, and Pauling, showing comparisons of humans and chimpanzees, humans and gorillas, old world monkeys and humans, et cetera, and showing the number of amino acid differences. This idea that the older the divergence, the more molecular changes will be observed between the two lineages. Now, another insight that came early in terms of the history of the molecular clock is that different proteins, different genes, evolve at different rates. And this is shown here with these different proteins, and shown here representing in an hourglass where the fibrinopeptides have a very fast rate of evolution. The hemoglobin and the cytochrome C have more of a intermediate or moderate rate of evolution, meaning that there's fewer changes as you go back in time. And then genes like histones essentially have a zero rate of evolution because they're highly conserved. So in a sense, the idea with the molecular clock was that if you can sort out this relationship between the amount of genetic change to the time since divergence, the promise of the molecular clock is that you could then estimate the ages for lineages for which you don't have fossil information. So all of these points in this plot, there is information from the fossil record on the time that the two lineages last shared a common ancestor. But the idea with the molecular clock is that even if you have comparisons for which there is no information about the age of their common ancestry given the fossil record, all you would need to do is to have this relationship sorted out and observations on the genetic divergence between the lineages, and utilizing this relationship, be able to estimate the age for lineages for which we don't have information from the fossil record. And very soon after the introduction of the molecular clock, it became apparent that different proteins have different rates of evolution. In a sense, there is no universal clock across all loci. So this is just diagrammatically representing the same idea, but now we're thinking about DNA sequence changes. And with the onset of genomic information, it is doable to apply molecular clock approaches among the most closely related species in the tree of life. Previously, with the hemoglobin, it would be very difficult with the amino acid sequences, it would be very difficult to distinguish the divergences at the time scale that we think speciation is happening. That is hundreds of thousands of years into millions of years, but certainly not hundreds of millions of years among the most closely related species. Now, it's, as we've shown, as we've talked about, so here are the amino acid changes. Here's the time since common ancestry, that different proteins have different rates of molecular evolution. And at least all of these seem to be clock-like, meaning that there is a relationship that can be discerned between the amount of amino acid changes and the time since common ancestry. However, here's a really interesting comparison with nucleotide substitutions. So here we're looking at the number of substitutions in the cytochrome C gene. That's the gene here, which we were looking at the amino acid sequences. And what we're seeing is that there are more nucleotide substitutions over a given time period than there are amino acid substitutions. Why is that the case? Why would you expect that for the cytochrome C gene in this case, for a given comparison, that there are more nucleotide substitutions than there are amino acid substitutions? Let's give you a moment to think about that. Okay, well, if you were thinking about the redundancy of the genetic code as to why there would be more nucleotide than amino acid substitutions, you're exactly right. And we've talked about this before, where mutations at different sites in the protein coding DNA sequences have different effects. So essentially, you can have a nonsense substitution where we're going from an amino acid to a stop codon. These obviously would be strongly selected against. You could have a change, any change, remember, at a second codon position will be a replacement substitution. It will change the amino acid. We call that a non-synonymous substitution. And then many of the changes at the third codon position and some of the changes at the first codon position you'll have a change in the DNA sequence, but you don't have a change in the amino acid. And these are called silent or synonymous substitutions. So the fact that you can have DNA sequence changes that are not changing the amino acid is driving that observation that there are more nucleotide substitutions for a given stretch of evolutionary time than there are amino acid substitutions. Now, the synonymous and non-synonymous substitutions are clearly evolving at different rates. So here we're looking at the number of nucleotide substitutions in years of isolation of different influenzaal viral lineages. And we can see that the synonymous substitution rate is much higher than the non-synonymous substitution rate. And even if we look at the different sites within the codon, we see differential rates of evolution. So as we would expect, given that second codon positions always result in a change of the amino acid, here we are, we're looking at sequence divergence versus time, that indeed second codon positions here in red have the slowest rate of evolution relative to first codon positions, and then third codon positions have the highest rate of evolution. And in fact, the rate of evolution at third codon positions is so high that the changes essentially become saturated, meaning that they cannot accumulate any more divergence because essentially there have been so many changes. And remember, there are only 3 other possible states for a nucleotide to evolve to. So in thinking about estimating divergence times, we want to be mindful of these differential rates of evolution in synonymous versus non-synonymous substitutions, and even the different codon positions within the protein coding gene. Okay, now another problem with molecular clocks is that we see that there are different rates of evolution among different lineages. So the idea of the molecular clock as introduced by Zucker-Kandel-Pauling was assuming that the rates of evolution of these genes was the same in all of the lineages. Well, that's rarely the case. And one way to investigate if rates of molecular evolution are uniform between 2 lineages is to use a relative rate test. And this is a very straightforward test. So what we're doing is we're asking, are lineages capital A and capital B, are they exhibiting a similar rate of molecular evolution? And the way we would do that is we would say, essentially the sum of branch A, branch C, and the branch of an outgroup here. So E represents an outgroup. So the sum of the branch lengths of A, C, and D should be the same as the branch lengths of B, C, and D. So you see that? So if rates of evolution, molecular evolution, were differing in these 2 lineages, one lineage would have a longer branch than the other, and we would be able to detect that in the relative rate test. Now, more often than not, rates of molecular evolution are not uniform among lineages. And when we are estimating the time-calibrated phylogeny, when we're estimating the divergence times, essentially we have statistical tools to account for the variation of rates that we may expect to encounter. And by using distributions of those expected rates, we could then accommodate for the rate variation that we see across the phylogeny. Now, that's the advanced phylogenetics course material for all of the EEB nerds in phylogeny nerds in the class. You'll be exploring these topics in later courses as you move forward your education here at Yale. Okay, now the rate of molecular evolution among these lineages, it's one thing to say it's not uniform. It becomes a really interesting question to ask why it's not uniform. And there seems to be a variety of reasons. So two reasons may be that, well, or two factors that seem to be associated with rates of molecular evolution are generation time and metabolic rate. So the idea is that lineages with high metabolic rate will essentially be producing a greater amount of metabolic byproducts that may affect mutation in and then increasing the nucleotide substitution rate, or increasing the molecular clock in a sense, those lineages. And also the idea that lineages that have a short generation time will essentially have more generations per unit time, increasing the probability of any given mutations from entering into populations. And a really elegant study that was done in the early 90s showed that if you look at the range of metabolic rate and generation time, you see a really interesting pattern. So all mammals are endotherms and that they're warm-blooded, but mammals exhibit quite a range of variation in metabolic rate and generation time. So whales here have a slower metabolic rate relative to these other mammals, and they also have a really large, they have a large body size, and they have a longer generation time than most other mammals. And what we see on the other end of the spectrum here is a mouse that has a relatively high metabolic rate, a low body mass, and a much faster generation time. And we could see this relationship between body mass as a proxy for metabolic rate and the rate of molecular evolution. So among endotherms, the larger species tend to have a slower molecular evolutionary rate than the smaller species. Within the ectotherms, that is the cold-blooded organisms, we see a very similar pattern in that the smaller bodied organisms have a faster molecular evolutionary rate relative to the larger bodied organisms. Now, also, overall metabolic rate dramatically differs between endotherms and ectotherms. And we see this, that ectotherms overall have a much slower rate of molecular evolution than the endotherms, okay? So generation time, metabolic rate may be driving the differences in molecular evolutionary rates that we see among different lineages. Now, we even see this in plants. This is a really elegant study that was done by Michael Donahue, who used to teach Bio 104 here at Yale. And at the time, his graduate student, Stephen Smith, who's now a professor at the University of Michigan, had shown is that the plant lineages that have very different life histories are exhibiting differential rates of evolution depending on the life history that the species is in. And the life histories I'm talking about are whether they're long-lived, forming trees, or whether they're short-lived, more herbaceous like plants. So in these 5 examples, in green, there's the phylogenetic tree for these 5 different groups of flowering plants of angiosperms. The green branches are the lineages and species that are herbaceous. The brown lineages are those that make trees. And you could see in most of these cases, the herbaceous things are not necessarily monophyletic, and the tree things are not necessarily monophyletic. And what these box plots are showing for each of these clades is the rate of molecular evolution predicted in the lineages that are tree-forming, brown, or herbaceous, green. And the herbaceous lineages are exhibiting a faster rate of molecular evolution than the tree lineages. So again, showing that life history and of the organism may be a factor in determining its rate of molecular evolution. Okay, so how do we in practice proceed? So let's say you're doing some independent research and you're generating phylogenies and you want to make a time-calibrated phylogeny. So if you go out and collect some DNA sequence data, either on your own or you download data from GenBank or download data from a genome browser such as Ensemble, you'll utilize the phylogenetic methods that we talked about earlier in the course, and you're going to generate a phylogeny that's going to look a lot like this, meaning that the branch lengths are proportional to the number of changes, the number of nucleotide substitutions that are optimized as changing on that branch. So we get a tree here where the branch lengths are corresponding to the estimated number of changes, and you could see that this is not clock-like, right? That this would fail a relative rate test if we were to look at the sum of this branch and this branch versus the sum of this branch and this branch. But basically what we do is we take this DNA sequence inferred phylogeny and we convert this to a relative chronogram, where now all of the living species are set at the same time, the present day, and the nodes and the branches we get are estimated either by stretching or compressing through either a strict molecular clock, where we're assuming that all lineages have the same rate of evolution, which, as you can see here, we would reject, or using a relaxed molecular clock approach. And this is what I alluded to earlier, where we have approaches, statistical approaches, to accommodate the anticipated or expected heterogeneity rates that we would find across the phylogeny. So let's say we've done that. We've taken our phylogenetic tree that we've estimated, and we've done these methods to essentially relax the molecular clock to accommodate the fact of our observation that these lineages are not exhibiting the same rate of molecular evolution. Well, if we do that, this is what we get. We get a chronogram here where the branches are drawn to reflect some aspect of relative time. And I say relative time because these branches and these timings are not calibrated with any external information that would allow us to have absolute time. In order to convert this tree from relative time to absolute time, we need some external information. And that usually comes from the fossil record. So let's think about that. How do we do that with regard to the fossil record? And it's very straightforward. It's very straightforward. What we do is, let's say we have these two lineages, all right? Let's call this lineage A and lineage B. In lineage A, we have no information of the fossil record, all right? So with lineage A, we're SOL, there's no fossils available. But in this case, with lineage B, we know, let's say we know that the oldest fossil that we can assign to lineage B, in this case, is 20 million years old, all right? Now, either somebody has told us that the fossil belongs to that lineage, or maybe we've done our own phylogenetic analysis of morphology to place that fossil at lineage B, okay? Regardless, let's say we're very confident this fossil belongs to lineage B and it's 20 million years old. If that's the case, and if we know that we've sampled well taxonomically that lineages A and B are sister lineages, if A and B are sister lineages and the oldest fossil of lineage B is 20 million years, then this split between A and B must have happened at some type before 20 million years ago. Now you might be saying, oh, Prof. Neer, now you can't tell me when that split happened. You can only tell me it happened before 20 million years ago. Yes, that's what we're saying. But think about it. The fossil here is providing a minimal age estimate for that node. We know it cannot be younger than 20 million years. So now that we have this minimal age constraint, now we could go forward and with our relaxed molecular clock approach here, we could go ahead and now convert the entire tree to absolute ages. So all we need is information for one part of the tree, and we could then estimate the ages for the whole tree. And as I said, remember, we said this must have happened sometime before 20 million years ago. And indeed, in this hypothetical example I've drawn out, indeed, the age of this node is reconstructed at some point that is older than 20 million years, but it's not younger than 20 million years. Now, when we do these analyses, often we're using multiple fossils. There are now very sophisticated algorithms and models that account for the expectation of sampling fossils in the fossil record, to where this calibration approach here is presented in a very straightforward manner, but there is a lot of sophistication that's available today for doing these types of calibrations. Okay, and always when we're estimating these chronograms, we have credible intervals around these divergence time estimates. You could think of a credible interval as the standard error in a statistical sense around these age estimates, but these are often Bayesian age estimates, so they have a credible interval, meaning that we have some idea of the confidence, the range of ages, in a sense that we accommodate in thinking about the ages of these different groups. Now, let me show you a really interesting real-world example. In our interaction sessions on Thursday and Friday, we had a couple of questions about mammal phylogenetics. So here's a great example where fossils have been used to calibrate the split between humans and chimpanzees at about 6.6 million years. And in fact, fossils from the same formation of 6.6 million years are also used to date the split between the baboons and macaques. Now, these are obviously different fossils, but just so happen that these important fossils are found in the same formation. Hence, that's why they have the same age of 6.6 million years. And in the case of the chimpanzee-human split, these fossils are resolved on the human lineage, not the chimp lineage. But based on these two calibrations, we could then estimate the evolutionary ages of the splits among all of the major lineages of primates. And you could see here that the split between the Western Hemisphere monkeys relative to the African and Asian monkeys and the great apes, the estimated age is about 43 million years. And you see these numbers in the parentheses. This is the credible interval that I was referring to in the previous slide. So in a sense, we would say the age of that common ancestor of the Western Hemisphere and the African and Asian primates is at 43 million years, with an interval of about 37 to 52 million years. So those are the range of ages that we accommodate given the uncertainty of these age estimates. And the really interesting insight into this time-calibrated phylogeny is that the splits among the major groups of the primates go all the way back to the Cretaceous. Remember, this is where the Cretaceous Paleogene mass extinction occurs. And this is contentious because there are no, not just primate fossils in the Cretaceous, there are no placental mammals in the Cretaceous. And yet the divergence times for many of these early divergences of placental mammal clades go back into the Cretaceous. Okay, so that's great. We can, in an esoteric sense, yeah, we can know how the ages of these lineages. What's the big deal? What are we actually using this for? So I want to switch gears a bit, and we're going to think about how uh, having a temporal understanding in the context of phylogenetics really allows us to investigate important questions in evolutionary biology. Okay, so this is one of my favorite group of studies. This is a group of studies uh, carried out by a really excellent evolutionary biologist who's at Dartmouth, Mark McPeak. He studies a group of insects that are related to dragonflies called damselflies. So you'll always note a damselfly because they fold their wings over their back, whereas dragon wings, the wings stay out. And you always find damselflies in riparian areas around rivers and streams. So I see them a lot in my work with North American freshwater fishes, and they're beautiful, beautiful animals. Now, in Mark McPeak's work on damselflies, a really wonderful byproduct of his work has been the generation of a lot of well-sampled species-level phylogenies for several of these closely related lineages of damselflies. And a really interesting pattern emerges that exemplified here with damselflies, but almost always, if you're working with species-level phylogenies, you're going to run into these patterns. You're basically going to run into combinations of what we would call depauperons, that is depauperate lineages, where you'll have a long bare branch in the phylogeny. So this is a chronogram. I'm not showing you the estimated divergence times, but this is a tree as a product of a relaxed molecular clock analysis. So you have branches that are long and bare deep in the tree, in that there are no other lineages along this branch, just this singleton species. So we may say that there are fewer species in this lineage than we would expect by chance alone. On the other hand, look at this group here, where in a very short period of time, we have the evolution and diversification of many, many species. Almost half of the species in this phylogeny are all in this very closely related shallow clade. This would be a rapid radiation where we're seeing more species that in a given time interval than we expect by chance alone. So when we're looking at phylogenies, what we're seeing all the time with these time-calibrated phylogenies, particularly those at the species level, is that there's differences in the shifts of, there's differences in the rates of diversification, as well as where shifts of diversification are happening among the different lineages. Now, for our purposes, diversification is the net result of speciation and extinction. So we're talking about diversification. We're looking at the present day biodiversity that is the result of speciation, the generation of biodiversity, as well as extinction that has culled some of that biodiversity, okay? So diversification is speciation minus extinction. Diversification is speciation minus extinction. Now, in terms of Depauperons, I wish we had more time to talk about this. It's something I find really interesting. And this comes to this shaky idea in evolutionary biology of living fossils. Living fossils as lineages that have exhibited very little phenotypic evolution in their history. So we could go back and find fossil horseshoe crabs that look just like living horseshoe crabs that are hundreds of millions of years old. We could find lineages that we thought were entirely extinct, such as coelacanths. These are all also called Lazarus taxa, where coelacanths were thought to have gone extinct 75 million years ago. It's about 10 million years before the Cretaceous Paleogene mass extinction event. Then in the late 1930s, coelacanths were rediscovered. Living coelacanths were discovered in South Africa. Their primary area of distribution is around the Comoro Islands in the Indian Ocean near Africa. as well as in the Indo-Pacific around Indonesia. Then there are lineages that are the surviving, singleton surviving and sister taxa to other very species-rich groups. So you may look at this and you may think, oh, this is a lizard. It's not a lizard technically. It's what is called a tuatara. It is the only living species in this major lineage that is the sister lineage to all living snakes and lizards. There are living fossils in plants. We already talked about the ginkgo and how there's essentially one species of ginkgo, and yet they are one of the 5 major living lineages of seed plants, right? And even within flowering plants, which contain about 257,000 species, there's a living fossil, if not many, but the one we'll point out here is Amberella, which is thought to be the living sister lineage to all other flowering plants. So what we see in the tree of life are not only that we saw with the damselfly, where there was that species with that long naked branch, that's probably an interval of about, I don't know, 3 to 5 million years. Here we're dealing with long naked branches that we're talking about hundreds of millions of years. So what's going on? So how do we get Depauperons? Well, the way we get Depauperons is thinking about the dynamics of speciation and extinction. And what we're showing here on this tree is if we're increasing the diversification rate, remember, the net of speciation and extinction is diversification. Where diversification is increasing, these are the hotter colors. Where diversification is slower, these are the cooler colors. Now, we can get Depauperons by really having low rates of speciation and really high rates of extinction, okay? So let's say this lineage was more species rich in the past. We have a high extinction rate that keeps lineages from accumulating, coupled with a low speciation rate that keeps lineages from originating, and you can get a Depauperon through time. Another way to get a Depauperon is basically to have essentially a flat extinction rate, but a very low speciation rate. And the way we can get these radiations where lineages that have more lineages than we expect by chance alone, that could come through a fairly flat extinction rate, but a very high speciation rate. So how we see biodiversity are distributed across the phylogeny is the product of differential rates of extinction and speciation. And there's some stochasticity associated with this as well. But in lineages where speciation rates are low, extinction rates might be flat, we will have Depauperons. If we have lineages where speciation rate may be intermediate, but the extinction rate's really high, we'll have Depauperons. So living fossils, there may not be necessarily anything intrinsic about living fossils. There may not be anything about them as to why they're losers in the evolutionary game of speciation, but it could be just different contingencies, similar to when we talked about Stephen Jay Gould's ideas associated with the Burgess Shale, that lead to the differential patterns of how biodiversity is distributed across the tree of life. So the takeaway here is what I really want you to think about when we were covering the different major lineages of plants and the different major lineages of animals. Often numbers of species were presented to you. This is not just for you to memorize for the sake of memorization. What I really want you to do is to be thinking about these mechanisms of extinction and speciation and how they may be resulting in the patterns of biodiversity that we see distributed in the tree of life. For example, you could take this line of thinking and start asking questions as to why there are 10,000 species of birds, there's only half the number of species of mammals, and there's order of magnitude fewer species of turtles and 2 orders of magnitude fewer species of crocodilians. And so we could think about how deploying different rates of extinction and speciation uh, will help us to explain those patterns of biodiversity and the disparity in the species numbers among major groups of plants and animals. Okay, now let's talk about something that's not extrinsic, something that is intrinsic. Are there aspects that lineages evolve traits that allow them to diversify at a higher rate than would be expected with lineages without that trait? So here is a really neat group of plants called the columbines that you find on the Pacific coast of North America. So if any of you are from California, you've probably seen these if you've been out and about hiking on the Coast Range. So the genus that we're thinking about here is Aquilegia, and the columbines have this interesting trait of these nectar spurs that provides a unique reservoir of nectar for different pollinators that can get into that spur. Now, the idea here is, do lineages evolve traits that facilitate the occupation of a new set of habitats or utilization of a new set of resources? And this idea is called a key innovation, a trait that allows the lineage to exploit a set of habitats and resources that other closely related lineages do not. So here with Aquilegia, we're looking at the species that have the spurs versus the species that do not have the spurs, colored here in pink. The species that have the spurs seem to exhibit a faster rate of speciation, more species generated here. Again, this is a time-calibrated phylogeny than the lineages that do not have the nectar spurs. And this is shown if we look at the columbine phylogeny in a slightly different way, and changes in the length and the color of the nectar spur has facilitated shifts between different pollinators that is argued to have driven their rapid speciation. The pollinators being long-tongued bees, hawk moths, which have really long tongues, and hummingbirds. And the different pollinators are indicated here with the different colors, that a shift to a hawk moth and a hummingbird pollinator is thought to facilitated the rapid speciation of those lineages. Now, key innovations, now we're going to go back to the origin of the idea of key innovations, which really came in looking at the relationship between plants and insects. Now, the idea is a key innovation can result in increased diversification by also reducing or slowing down the rate of extinction. Remember, diversification is the net of speciation minus extinction, okay? So many plants produce latex that is exuded when the plant is damaged. And this is to protect against herbaceous insects. And if you look at the number of living species in these different plant lineages, those that have nectar versus those that do not, and the nectar, that have the latex versus those that do not. So we have the ginkgo with one species, its sister lineage, the conifers, the pines do. There is a dramatic difference in species diversity. Here we have the fig family of plants and versus the elm family of plants. Excuse me. And again, we have a really big, appreciable disparity in the number of species. So the idea here is that perhaps by having this innovation of the latex canals has reduced extinction and facilitated the accumulation of greater species diversity in the lineages that have the canals. Now, a key innovation could also result in increased diversification by increasing the speciation rate and decreasing the speciation rate. And some examples that have been proposed that are doing just this is herbivory in insect clades. So again, just like with looking at the latex, we see that the ladybird beetles that are not herbivorous have only slightly less than 10,000 species versus weevils have 130,000 species. And again, over here, we have caddisflies, which spend most of their time as aquatic larvae. They are not herbivorous, but whereas their sister lineage, the Lepidoptera, the moth and butterflies are, they have 140,000 versus 7,000 species. So the fact that these herbivorous insect lineages are more species rich, it's thought that this is a key innovation, herbivory is a key innovation that would have driven the speciation rate as well as lowered the extinction rate. Okay, now another way that lineages can exhibit or respond to and exhibit high rates of speciation is when lineages occupy island archipelagos, and islands will initiate radiation because there are a lot of open niches, available habitats and resources, and these lineages, in the case of Galapagos finches, begin to occupy these different habitats. They're exhibiting ecological differences among the closely related species that are associated with utilization of different resources and habitats on the islands. And indeed, if we look at the phylogeny of Darwin's finches here at node 2 versus all of the other birds that are not on the Galapagos but are closely related to Darwin's finches, this is a time-calibrated phylogeny, and we see indeed that Darwin's finches are exhibiting a shift to a rapid rate of diversification relative to these birds that are not on the islands. So it's not a key innovation in this case, it's a key opportunity. The opportunity, in a sense, of making it to the islands and occupying the different niches, and then evolution taking its course and having a high rate of speciation. Now, one example that, another example of that blurs the line between key innovation and key opportunity is a really interesting group of fishes that I've studied quite a bit. These are called Antarctic notothenioids. So what we're looking at here is a species, Trematomus borchgrevink, that this fish is living in the interstitiaries of the sea surface ice at McMurdo Sound in Antarctica, okay? Now, if you or I were to, with our hand, by the way, this water is cooled to negative 1.86 degrees centigrade, so well below freezing, but freezing point is depressed because it's seawater. If for some reason we were to try to touch this fish and our hand touched those ice crystals, because the water is super cooled, our hand would immediately freeze. Now, this fish has the same osmolarity as you or I have. So why is this fish not frozen solid? This fish is not frozen solid because it has this really interesting simple protein called the antifreeze glycoprotein, which is an alanine-alanine-threonine repeat. It's a triplet that's repeated hundreds of times, scores or hundreds of times. And by the way, there's different lengths of these repeats. It has a disaccharide chain. And what these antifreeze glycoproteins do is they glom onto ice crystals. So this fish is actually sequestering ice in its body, but the ice crystals are not growing because they're covered with the antifreeze glycoprotein. Now, this is a really poorly drawn phylogeny of fishes, of teleos fishes. But what's interesting about this phylogeny is that the Antarctic notothenioids are shown here. They have antifreeze glycoprotein. Another group of fishes that are found primarily in the Arctic the cods, COD, they also have an antifreeze glycoprotein that they evolved independent of the antifreeze glycoprotein we have in the notothenoids. And these lineages are not closely related in the teleos tree of life. Now, work that I've done in my lab, I've taken advantage of the fact that most notothenoids, I'm sorry there's not a good common name for these species, but if you've ever heard of Chilean sea bass, which you'll often find available on menus in restaurants, that is a notothenoid. Most notothenoids live in the Antarctic. I'm showing you a time-calibrated phylogeny here for the notothenoids. The blue lineages are those you find in the Antarctic. We're rather fortunate that the closely related lineages that are related to the Antarctic notothenoids are not in the Antarctic. They're in extreme cold temperate southern habitats such as extreme southern South America, southern Tasmania, southern New Zealand. So these are lineages that have never been exposed to the deep freezing conditions of Antarctica. Now, what I'm showing you here, this is geologic time, and this is a reconstruction of paleo temperatures. So what happens at about 35 million years ago, the world went from a very warm condition to a fairly cold condition. This is called the Eocene, Oligocene thermal maximum. So at that point, the world starts getting cold, and what we see with the temperature reconstruction, that about 10 million years ago, Antarctica really started to get cold. So the question we had in thinking about the evolution of the antifreeze glycoproteins, where are they evolving in the history of the notothenoids? And indeed, what we see, if we think about reconstructing the presence of these antifreeze proteins in the common ancestors of our reconstructed phylogeny, what we find is that the antifreeze glycoproteins evolved at some time, either coincident or after the onset of the cold conditions that characterize Antarctica. So was this really a key innovation, or was it really a key opportunity? So what we know about the Southern Ocean prior to the onset of cold conditions, because of the fossil record, it had a very diverse and cosmopolitan fish fauna, meaning that there are many different lineages of fishes living there. However, today in the Southern Ocean, there's really only one lineage of fishes that occupy the Southern Ocean, the notothenoids. So the idea is that just like with the Darwin's finches, coming to the Galapagos Islands and finding no other birds there, that they're able to take off and occupy those different habitats, the notothenoids found all of those habitats cleared of other fish lineages, and they were able to diversify to fill all those other niches. And indeed, what we find is after, it's about 10 to 15 million years after the origin of the antifreeze glycoprotein, that we find Antarctic notothenoid lineages that exhibit high rates of speciation, high diversification rates. So it seems that the antifreeze protein is an innovation that allows them just to live there, but the actual clearing of the niches of those other fish lineages is what was really important in the incredible species diversity that we find in the Antarctic notothenoids today. So in a sense, the notothenoids had a key opportunity to exploit those different habitats. Now, probably the most spectacular rapid radiation in the tree of life are the cichlid fishes in the rift lakes of Africa. Now, these lakes are all connected by rivers, well, not connected to each other, but they're basins that there's rivers that drain into these lakes. And these rift lakes were formed somewhere about 50 to 30 million years ago with the rifting of the East African area, all right? So these lakes, the three that we're going to talk about, Lake Victoria has 500 species, Lake Tanganyika has 200 species, Lake Malawi has 500 species. Now, the fishes of Lake Malawi and Lake Victoria are each monophyletic. meaning that all of those species look like they descended from a common ancestor. Now, Lake Tanganyika is not monophyletic, but a lot of the lineages clearly diversified into Lake Tanganyika. Now, check this out. The age of the lake basin for Victoria is no more than 15,000 to 100,000 years old, and less than 5 million years for Lake Malawi. So in these short period of time, both Victoria and Malawi have diversified to 500 species. Now, the Rift Lakes are island-like, but the radiations have happened in the individual lakes. How did that happen? So the idea is that if we dial into these different cichlid lineages, what we end up finding are lineages that are diversifying first on what type of habitat they're occupying, rocky habitat or sandy habitat. Then in both of these instances of rocky and sandy habitat, they're diversifying with regard to what they're eating. And we see this reflected in their oral morphology. So diversifying rocks, having different oral morphologies that are correlated with eating different things. Then within each of these oral morphologies, we have diversification of color pattern. And there's parental care in cichlids. And so male-male interactions and female choice are very important in setting up reproductive boundaries between closely related species. So it's a set of nested replicated radiations involving habitat, diet that's reflected in their oral morphology, and then secondary sexual characters. That is assortative mating that is related to behavior and color patterns among these different closely related species. Now, what's really exciting is that there's really incredible parallel evolution among these different lakes. Now, all of these Lake Malawi species are more closely related to each other than they are to anything in Lake Tanganyika. And what we see is that in Tanganyika, Malawi, we have this parallel evolution of very similar phenotypes that are associated with utilizing very similar habitats and resources. And so when we talked about Stephen Jay Gould's idea of contingency versus adaptation in evolution, this would be a counter argument to contingency, that given these lakes, which are different, have their own evolutionary histories in these lakes, the cichlids have their own evolutionary, the different cichlid lineages have their own evolutionary histories, that evolution is resulting in very similar outcomes. Now, this idea of replicated evolution, you're familiar with by engaging with the reading of Jonathan Losos and his work with anolis lizards. Sorry, this slide is out of date. He is at Washington University in St. Louis, no longer at Harvard. And as you learned by reading his work, the paper looking at replicated evolution in the Caribbean, there's about 390, maybe 400 species of anoles, and more than 150 of those are found in the Caribbean. And these island lineages are thought to have been initiated by just a few mainland colonists. And the anolis exhibit these different ecomorphs where they're occupying different habitats and trees and on the ground and in shrubs. And the morphology we see corresponds to these different habitats that they're utilizing, hence ecomorphology, ecomorphs. And in the study that we read, what we see, or what Losos discovered, is that particularly we look at the large islands of the Caribbean, the same ecomorphs as colored here by different branches of the phylogeny have repeatedly evolved on the different islands. So it's not as if all the crown giants form a clade, all the trunk crown form a clade, that on each of these islands, the islands in this case are forming the clade, and in each of these clades, the different ecomorphs are evolving. Okay, So in thinking about rapid radiations, some of them seem to be facilitated by key innovations, that is novel traits, others by key opportunities, movement to new and unoccupied territories that both can initiate cascades of evolution. Now, we didn't get into the concept of adaptive radiation in this course, but some of these are adaptive radiations where they're generating large magnitudes of disparity among closely related species. They're occupying multiple distinct ecological niches. But some radiations can be rapid. Some radiations can produce a lot of species that look very similar to each other. So think of this would be the opposite of an adaptive radiation. And radiations are sometimes repeated in different areas. Think of the different rift lakes in East Africa, as well as different islands of the Caribbean. Okay, from this lecture, you should be able to think about relative versus absolute time in phylogenetic trees, the molecular clock, how we test it, and why it may not be uniform. That is why there is no universal molecular clock. You should be able to think about the natural history of genes in the sense of synonymous and non-synonymous substitutions and variations in nucleotide substitution rate among different codon positions. You should think about how we can place fossils into phylogenies and how this allows us to calibrate relaxed molecular clock analyses. You should be thinking about why evolution produces lineages that we call Depauperons, that they're depauperate lineages. And you should think about rapid radiations and their possible causes that include key innovations, geographic opportunities, key landscapes. And we could think about, sorry, we didn't talk about Hawaiian radiations, ignore that. We talked about Antarctic fishes, African rift lake cichlids, and Caribbean anoles. Okay, Bio 104 students, I will be preparing to record our final lecture of the semester. So until then, be safe and be well. Bye.